I always have what's I call the grandma test.
Yeah.
Which is like, you put your own grandmother on this trial.
Yeah.
And if you wouldn't, then at least for me, then you really shouldn't be developing that molecule. So I would just keep that standard for myself, at least.
100% agree.
Okay.
Normally I ask people to give a background on themselves, but yours is too long. So we'll just hit it as we go.
Okay.
Well, what's your 1-minute version of your background?
Let's see, I'm a physician scientist that started out in academia, went to pharma, and is now in biotech.
Yeah. So what made you want to leave? I mean, you were running a lot of cool stuff at Vanderbilt, and then you decided to go to the pharma side at Roche. What made you want to make that jump to the pharma side?
Yeah, so in my academic career, so I'm a physician scientist by training. I saw patients, had a lab, helped to run trials. And during that academic career, I was focused mainly on targeted therapies in lung cancer.
Yeah.
And I also helped other companies develop their molecules.
Yeah.
So, for example, I helped AstraZeneca develop AZD9291, which became osimertinib with Tagrisso. Also helped a small company called Discovery develop another molecule, which actually was also approved in sartanib.
Yeah.
And then I got headhunted, and most of the headhunting offers I had turned down.
Yeah.
But this one was from Roche, and Roche is very scientifically strong.
Yeah.
Particularly on oncology. It was to head up oncology. And I did agonize for a long time, but after I met the folks there and weighed the pros and cons and thought about what I would do in the future in academia versus in industry, I decided to make the plunge. And the main reason was that I wanted to be able to do drug development full time.
Yeah.
And then ultimately have impact on patients, maybe hundreds, thousands, maybe even more than that. Whereas I do miss seeing patients one at a time. Yeah. But the impact from drug development, it can be much greater.
So it was like a trade-off of, you have a 100% chance of helping a few thousand a year versus like I have a 20% chance now of helping millions.
I don't know if it's 20%. It could
well, you know what I mean.
Yeah.
I'm saying with your selections, it was a bit higher than the average.
Yes, my PTS. My probability of technical.
So what do you think was most different about pharma relative to what you expected when you were going in?
Yeah, so I found that the science, I think, is equally the same quality, like the quality of the scientists and the stuff that's being done. But obviously in pharma, it's much more directed. Yeah. Whereas in academia, you can really follow any idea what you want. Yeah. And you have more freedom to do that. The other main surprise was the number of people in pharma that are experts in diverse areas. Yeah. So as an academic, I usually interacted with one or two people in the pharma company, like the key scientists on the project or the key clinical person. And then I got to Roche in Basel, Switzerland, and I was like, wait, there's an expert for formulation, there's an expert for, whatever topic, toxicology, dosing, and so on and so forth. And just sort of realizing that, wow, there's all these scientists that really do amazing work, but in particular areas, and they all contribute to the development of the molecule. Yeah. It's sort of like a tertiary, when you're in a tertiary hospital versus a primary care hospital. Yeah. And in a tertiary care hospital, you have just levels of expertise.
Well, I think there's also I was coming at it from the student angle, but I think there was this misconception that, we the academics are the smarter ones, and then the pharma and biotech people are slightly stupider on average. And then you actually go, and you're like, they're actually pretty damn good.
Yeah, I think growing up when I was a fellow, I probably had that attitude. Yeah. But I think, in today's world, you can't be a successful pharma company without having cutting edge scientists. Oh
yeah. No, it was very, I think it was just like one of those weird misconceptions that the academic side would nurture for some reason.
Yeah. Well, like in oncology, not with no disrespect. Yeah. But in the 80s and 90s, a lot of it was about developing chemotherapies, right? Yeah. And then a lot of it was empiric.
Right. That's
like
true.
A mechanism based thing. Yeah. So I think some of it came from that baggage.
At least in
oncology.
Yeah, because they're like, we basically have these poisons. Let's mix them in cocktails and see what happens.
Yeah. And I even know the cancer biologists would look down on the clinicians and be like, you just are giving poisons, whereas we're figuring out the mechanism. But now, with targeted therapy, and you'd have to totally understand the mechanism. So
yeah, that makes sense. So what does the job actually entail when you're on the pharma side? So let's start with head of oncology. what's the day like? What are you up to? Especially, what are the things that you think are non-obvious to people?
What are non-obvious? Yeah, well, I mean, every day is totally different.
Yeah.
You can be interacting. Well, I was the head of oncology for Roche for 4 years, the Roche Pharma Research Early Development side, and then I was head of the whole period. So in oncology, I mean, you can be interacting with scientists one day, you have to be interacting with the clinicians the other day, you're looking at data, whether it's clinical or basic science.
Yeah.
You have to figure out what the strategy is.
Yeah.
I was fortunate to hire folks like Christian Rommel and Friedrich Finkenstein as my heads of discovery and clinical, respectively. And we had to, I think one of the things you have to do in drug development is envision the future.
Yeah.
And not only envision the future, but like figure out like if I'm working on something today, is it still going to be relevant by the time it gets approved in say 5 to 10 years?
Yeah.
5 if you're lucky, right?
Yeah.
Probably more like 10. So a lot of that strategy is really important.
And what's the feedback loop like between kind of your vantage point versus, let's say, pharma's obviously an organization with many stakeholders, like the commercial side.
Yeah.
So you're like, oh, I think if we want to have maximum impact, let's do X. And then commercial brings in a perspective like, oh, actually, we think Y. Like, how does that work there?
Yeah. So I was fortunate that my CEO at the time was Severin Schwan.
Yeah.
Who would actually, and this is not a secret because he would say it in public too, he would tell the research guys not to listen to the commercial guys.
Okay. Which is different from the other pharma, you feel?
Well, let me get back to that.
Yeah. Okay.
So his point was that he actually had a famous laminated slide in his office and would say like, well, look, on the top of these are the probability of technical success of the molecules in development. On the bottom are the loss of exclusivities, and 90% of the data on this one slide that I've laminated is wrong. And his point was that, like, MabThera, which is a CD20 antibody for lymphoma, was originally projected to have like 200 million of sales, and it became, I think, a $7 billion blockbuster. Conversely, there were other medicines that were predicted to have billions of sales that didn't even make it.
Yeah.
So I think his point was very early on in the life of a molecule, it's hard to tell how big of an impact it's going to have.
Yeah.
Now, there's some grains of salt with that, which is like if you're going after a genetically defined disease in a rare disease population that's only 1 in a million, you might know that it's only going to be 1 in a million.
Right.
But if you're developing something like CD20 for the first time, an anti-CD20 antibody, it might work in one area, but then you don't know that's eventually going to work in, say, 10 different areas or something like that. So I think his point was follow the science. At some point, you do have to make a commercial decision. Yeah. Because in a big pharma company, a molecule should bring in enough revenue. Get into the math about that if you want. But early on, hopefully you just do the best science and follow that.
Yeah. Well, actually, a little bit of the math might be helpful for folks just to get a sense for like, what is this really tough equation people are trying to balance? Because I think from the outside at superficially, they're like, oh, it's 90% margin on these drugs. Like you're selling it for $200K. It costs you sub $10K to make it, like per COGS, but they don't understand all the failure rates and everything else that goes on. So what's your perspective on the math there?
Yeah. Okay. And then I want to come back to the commercial thing too. Yeah. Well, first I'm finishing on the commercial. Yeah. There are now shifts for some companies saying like, you can't even work on a molecule and discover it unless it's going to have more than $1 billion potential. As if you could predict that at a very early stage. So anyway, so to come back to the math. Yeah. So,
sorry, because you got me going now.
Okay.
Do you think, like, and maybe this is the wrong perspective, part of me has always thought of like, in pharma, commercial is closest to the money, let's call it. Whereas let's say in biotech, it's actually research that's close, because the money is the investor money and it's based on R&D data. No one's actually selling anything. So like R&D can drive it more. Whereas commercial side, it's like, if they spent $100 million and increased sales by 10% across the board, that is worth it all day, every day, versus like taking a punt on another drug. So is that where that influence comes from, in a way, that they're most proximal to what the investors and the headlines care about? Whereas in biotech, it's actually R&D that's most proximal to it.
Well, here's the way I think about it from a math perspective. So I had $1.2 billion a year of budget. Yeah. And on average, over 10 years, over 5 years, I was supposed to hand over 10 molecules to late stage. Yeah. So if you do the math on that, that was when I was head of period. You do the math, then it's 2 molecules per year on average.
Yep.
1.2 divided by 2 is 600 million.
Yeah.
So I'm spending basically 600 million per molecule to hand it over to late stage.
Yep.
And then when late stage takes it over, they need to probably spend maybe 100, 200 at least million to do the phase 3.
Yeah.
And maybe sometimes more. So by the time that molecule is approved, you spent like $1 billion. It sounds crazy, but that's what it is. But the 600 million per molecule that you hand over also takes into account all of the other molecules that potentially didn't make it.
Yeah.
Now, of course, if we had a higher success rate, then, you know, the number would come down. But on average, that's the way it is. So basically, if I'm spending 600 million per molecule, and my molecule eventually, when it's commercialized, doesn't make 600 million, I'm losing money for the company.
Yep.
And so over 10 years, I would have lost a lot of money. Right? So that's sort of where I think the blockbuster number comes from.
And what do you think is the greatest source of inefficiency of that? Because obviously there's the attrition rate of any molecule going in, but why is it that it's, you know, for 1.2 billion, you're only going to get 2 molecules, whereas it's not 4? Like, what would it take efficiency wise?
Yeah, I mean, I think if you only work on validated targets and you knew all the biology and you knew exactly which patient populations to go into, obviously you could have a much higher, what we call PTS, probability of technical success. Yeah. But you'd probably also only be making me too, me better, maybe even me worse molecules. Yep. So I think you have to have a mix of what you think you can improve on that are established targets, and then you do have to work on some novel targets. Now, you have a portfolio in pharma. Right. So I don't think you'd work on totally all novel targets because you might produce nothing. Yeah. But, you know, it's really interesting. As you probably know, every molecule that ever gets approved has to get to this intersection of biological understanding, clinical understanding of where the molecule might be relevant, and then technological advancement to make that target druggable. Yeah. And getting to that sweet spot, the Venn diagram, is not that easy. Right. Particularly for new targets. Yep. And so for pharma, if you really want to, well, and even biotech, if you want to be successful in a short period of time, it's much easier to go after a validated target than a unproven target.
Do you think, I know this is like somewhat tangential, but the incentive for making Me Too molecules, how much of it, or what do you think would remove more of that incentive? Because my impression is that it's mostly because, especially US commercial, you can have many drugs that are otherwise basically the same thing coexist and they will be commercially worth it. Whereas in other jurisdictions where someone just decides, hey, we're just buying this one and the rest don't have a play, that really kills the incentive for Me Too drugs. So is that what drives it, or what do you think drives it? And if we got rid of the Me Too incentive somehow, would it actually bias towards a lot of new generation molecules?
Yeah, that's a tough one because the challenge on the novel targets is, as I mentioned, it can take a long time. Right. And so if your horizon is 20, 30 years, or you have a long, investors that are in it for the long haul, then I think you'll be okay. Yeah. But if you have short-term investors, or if you're in a pharma company that has a patent cliff coming, and you need to generate that revenue quickly, otherwise your stock will drop and so on and so forth, then you do have to go after these probably more clinically validated. Or clinically validated can be that there's a molecule approved already, or it could be that your competitor's working on something and in a phase 1, like they showed like amazing data, and then everyone jumps into that bandwagon. Yeah. Now, what is interesting is you've probably seen some of these analyses. Now there's more herding, so to speak, than at least in the last 20 years. Yeah. I think people analyze the big pharma pipelines. Yeah. And there's a lot more crowding now on the same targets rather than, I think, 20, 25 years ago.
Because of the dynamics you mentioned, that like nearer term, it's more likely to yield a hit.
Yeah. Now, of course, it also, the market has to be big enough, right? So GLP-1, we see that there's a ton, but that market's expected to be $120 billion. Right. If unfortunately the commercial reality is that if it was a market of 200 million, I'm not sure you'd have so many people for sure getting into that space.
So with all these dynamics that you were balancing, what was the most difficult part of the job to you?
I mean, I think it's an amazing job. It's really fun. There's a lot of privilege to be able to do that. I think, you have to deal with a lot of stakeholder management in a big company. There's lots of different people. So even in your own organization, like there's different functions. Yep. There's the therapeutic area group, then there's therapeutic modality group, the pharmaceutical science group. So getting everyone aligned on the same vision and so forth. And then transferring over to late stage, you have to get a whole bunch of other people up to speed and convince, whether it's worth investing or not. And then having a large organization, that you have a lot of human resource topics to deal with as well. Of course.
Now, what were the aspects of this that you saw that you feel are most different from what people expect? Like even within the industry, everyone has their own like image of what someone like you does. But what's the aspect that you think is most different from what they think it is?
Oh, that's an interesting question. I mean, I think the higher up you go and more responsibility you have, maybe the less close you can be to the science. So that's one thing. Let's see what else. I think the main thing, so also going from academia to industry. Yeah. I mean, academia in my mind is a lot more about individual accomplishments and what have you done versus pharma and biotech, which is a lot more teamwork.
Yeah.
And the way I sum it up is in academia, I probably had to update my CV like every week. So it's like, what was the latest?
Senior author on this one.
Exactly. Yeah. And then, who did I train and so on and so forth. I have to submit my NIH CV every
Biosketch,
Biosketch, yeah. Whereas in pharma, I don't think I updated my CV for like 4 years.
Yeah. No, I mean, I definitely saw the team sport elements of it because I think that's one thing. Speaking of it from like the junior scientist side, that I think some scientists like end up hating academia for the like very individualistic, somewhat cutthroat at times. Because like if you're first author versus second author, that can be everything in certain situations, and so it's very non-team oriented. And then like industry is like, all right, we're all going to sink or swim together.
Yeah.
This molecule hits, everyone's going to be happy. If it doesn't hit, we're all screwed.
Yeah, exactly.
Obviously with all these different stakeholders involved, there's a lot of political pressures within a company, political pressures. How often did you feel like there were situations where there was political pressure to advance a drug that in your heart, you're like, no, I really wouldn't bet on that one. Or kill a drug that you're like, this one's going to be awesome. This would be one of my 2 for the year, but it gets shot down. Like how often was that a problem and how does one navigate it?
Yeah, the shooting down one, there was a particular molecule that I was working on that I won't name it, but it was what molecule. It's a target I've worked on in the past.
Yeah.
So you can guess. But this mechanism of acquired resistance was coming out to existing therapies.
Yeah.
And it wasn't clear when the initial data came out how frequent that resistance mechanism was going to be.
Yeah.
But by the time we got a small molecule to overcome that mechanism of resistance, it was a small fraction. And there I would have to say, unfortunately, and it pains me to say this, but I did have to take a commercial consideration and say, this is a great, well, the molecule had some liabilities also, but given that and then the tiny patient population, it was hard to justify. And I really felt bad about it, because in academia I would never do that. But I had to do that because it was right for the company.
And are there constructive enough mechanisms for these things to find another way to live on, or do you feel like they just like go on the shelf?
No. So those kinds of molecules can be licensed out, and then there's different size companies that are fine with different levels of revenue. So a big pharma company will need obviously a lot of revenue from its molecules, but there are mid-size or smaller size that are fine and happy with less revenue. So I do think in the long run those molecules will make it. And so hopefully in today's ecosystem that's possible. The other thing that I also sometimes got frustrated with is, because I'm a big proponent of personalized medicine, particularly in cancer, and some of the mutations get very, very small. The frequencies. And the issue is, during phase 1 and phase 2, a lot of academics want you to test your molecule in these specific small subgroups. But it becomes challenging, right? Because you need the biggest indication initially for late stage commercial guys to accept it. So I'd have to hold off doing those studies, but then with the promise that once it's approved, from a life cycle management perspective, those studies can be done. So it's not like it won't ever happen, but it just may not happen now. That was another painful thing from going from academia to industry.
And actually, one other curiosity that I just thought of is, because you were head of PRAD and then like at Pfizer, you were chief development officer. Was there a disease area that you thought the dynamics there was just way different from the other ones? Like, I'm just curious because I'm at least very used to oncology, but.
Oh yeah, I mean, I guess the way I used to think about it is what therapeutic areas are like precision oncology. You have maybe a genetically defined patient population. You know exactly which patients you want to go after. You have a therapy that hits that with a good therapeutic index and so on and so forth. Meaning like the mechanism is well known. And so that would be like, like precision oncology would be infectious disease where you know what
virus you're hitting or bacteria you're hitting.
Or maybe some rare disease where, again, the genetic situation. Then there's the flip side, which is you have no idea what the mechanism is. And so on. And so then you have to decide, like, how valuable is this, how worthy is this program moving forward?
Because it's like so much more uncertain what's going to happen.
Now, I do think having trained in oncology, as you know, we have concepts of tumor initiation, progression, and maintenance. And that's very helpful. It's like what genetic lesion or what happens to make the tumor start in the first place, then what makes the tumor continue to grow, spread, and then what is needed. The initial mutation that you had may not be needed later on, right? And these are all concepts that are really important for drug development. And I do think some of the other therapeutic areas don't have the same level of understanding, with no disrespect to those areas.
No, it makes sense. I think some of those just like might lend themselves less to it, or it's too complicated to figure out.
So from a portfolio perspective, then you sort of have to balance.
Because I could see like autoimmunity being one of them. That's like some of them are so freaking hard to figure out what the hell is.
Parkinson's disease.
Many have tried and failed. Like one interesting dynamic I think that you had to live with is this aspect of you, for example, as head of R&D, would make certain decisions that will not have readouts until way after you're probably gone. And also you're dealing with the consequences of decisions made before you that you had nothing to do with, but they like have their readout under your watch. What's that like? Like, how do you deal with it? Because I could see it being like your fault, even though it wasn't. Or your greatest work won't be known until.
Unfortunately, if you're in the leadership position and it happens under your watch, it's your problem. Or it's either you're a hero or it's your challenge. But I think the best advice I got from someone when I first went to Roche, actually the chief legal counsel, was like, you're only as good as your predecessor at the first 3 to 5 years or so. First 3 years, maybe. And then after that, sort of it's you. And your decisions. And stuff like that. So I think in pharma and drug development, you have to realize that you're on a relay race. You'll be there, but you have to make the best decisions during the time that you're there. Hopefully you build a pipeline that will produce when you're gone. And then hopefully the person that was in front of you also did the same. But as you know, probably, when you take over as the new head, often you do a review of the portfolio. And many times you end up killing a bunch of stuff that is unlikely to succeed.
And then the prior head's like, God damn it, that was my pet project.
I mean, a lot of times it's obvious, but you should stop. And coming back to, what's hard is like, I mean, I think a lot of teams do get invested in their programs. Some might have spent even 5 to 10 years on them, and so can become biased. And so somebody has to take that decision. And I realized actually as the head of the group that even my therapeutic area heads, who I thought were as unbiased as possible, even they had to like represent their group. So on and so forth. So actually it was part of my job to be killing programs. I mean, if I didn't kill them, who else was going to do that?
Then you were just like going to drown.
And killing them, frees up the resources and the people to work on stuff that's more likely to work.
And maybe a weird question, like, I don't know, how much contact you would typically have with other heads of R&D, but I'm curious across, because I mean, whatever, we're dominated by 10, 20 pharmas total. How often do you think the heads of R&D actually agree with each other, but the portfolios looking different is due to other forces and other dynamics, or versus like these people actually have very different views on what should be done?
Oh, I mean, I think when you go to a place that you have to really play to the strengths, right?
Yeah.
You can't just go to a place and say like, I want the other company's portfolio, right?
Yeah.
If you have no experts in diabetes or oncology, it's not like you can build it from scratch overnight. So I think when you go to a company, you're going to definitely inherit what's there already.
Yeah.
Then you got to assess like what are the strengths and weaknesses, and then what are areas that you could potentially go into, but not like over 20 years, that you could do in a reasonable amount of time. So I think obviously the clinically validated targets people will accept, but there are still debates on like, should we be going after, so like in Alzheimer's disease, A beta has always been a very controversial topic. Yeah. Even though there's 2 medicines approved now, these antibodies, some people still feel, that the improvement is not huge. Yeah. And so you may still have people say like, I'm not going to get into that space.
And like touching on the dynamics with biotechs, I mean, what do you think pharma is good at when it comes to innovation on new drugs versus what do you think biotechs are good at?
Yeah, that's a really good question. So now that I've been in biotech, I think pharma is good at actually, if their leadership is there in terms of long-term investment. Mm-hmm. So really being patient about some breakthrough molecule development. Yeah. And the examples of that would be like emicizumab, which is a bispecific molecule antibody for hemophilia A. Yeah. Where one arm binds to factor 9, another one binds to factor 10, and it bypasses the need for factor 8. Yeah. I don't want to get into too much detail. Yeah. But Chugai screened like 44,000. The scientists at Chugai screened 44,000 bispecific constructs. Yeah. And it was like over a period of probably 10 years before that molecule came into development. I think VCs would probably, at least today,
they would not do that.
No question. Have a much shorter horizon where they want their return. Yeah. And so that's unlikely. Another example is actually from Roche Purette was the brain shuttle, trontinemab, which is actually going to phase 3. It's A beta that you're talking about. But it took like 10 years of development to make a brain shuttle. Okay. Now, there are some biotechs that are doing that now too, but the initial investment, now that it's been shown that the brain shuttle potentially is more effective, now a lot of people are getting into there. Yeah. So I think some of those long-term projects, I think are best incubated in pharma. Yeah. And then biotech, I think it really can fund a lot of cool innovation. Yeah. It seems that biotech is often very close to the academic universities. Not that big pharma is not, but the VCs have really gotten into the space and licensing out and trying new promising technologies and therapeutic modalities and so on and so forth. So I think that's really good. Mm-hmm. I do. Right now, we're in a period, I think, where a lot of the VCs prefer a closer to near-term readout. Yeah. So one molecule is closer to clinic and so on. Yeah. But we do need to have investment in the earlier space as well.
And do you think like the cost of getting from, let's say, an idea for a molecule to first clinical proof of concept is lower and the timeline's faster for the biotechs relative to the pharmas, or do you think that's not necessarily true?
Well, again, let's see. Let me think about that one. Well, I think because biotechs tend to be smaller and have fewer people, there's just a lot less networking that's needed, and decisions can be made.
And they have the constant fear of death, so that definitely keeps them moving.
Yeah, for sure. Existence could be gone if the molecule doesn't make it.
There's a tiger chasing you, you're going to keep moving.
Yeah. Whereas in pharma, obviously, if your project is dead, then you get assigned to another project. Yeah. So I think in pharma, if you have the vision and the leadership that will support long-term, projects, then that's great. And then they don't have to rush as much. They have the time to really make, these breakthroughs. Yeah. Although even with emicizumab, coming back to that molecule, at one point, the leadership said, like, you have a year to show us that you can keep going. Yeah. And so you just still need to have deadlines in pharma as well. Yeah.
And what do you think leads to pharma doing, what feels like relatively few biotech deals, but when they do it, they appear to be paying such a significant premium relative to what the financial investors would have done? Yeah. So like, what do you think that dynamic is?
Well, I think there's several things going on, right? So in pharma, you have your own portfolio already. Yep. So sometimes you'll have what's called the not invented here syndrome. And that's really important to have your group culturally say that like innovation can come from anywhere, whether it's internal or external. Sometimes you will have to make trade-offs. So if you go for an external molecule, then it could impact internal people. So you have to, it's just a consideration. That's all. And then the other thing is it's always easy to say, yeah, your idea is really cool. I think it's promising, but show me more data. Yeah. And
no one's ever said that before to biotech.
No. And you're willing to pay more at that. I think it's just the uncertainty. Right. But you would think that pharmas would want to invest really early. So that they could get it cheaper, but that doesn't always happen. I think they're more willing to invest in my experience, in a platform that looks really promising. That may potentially open up a lot of avenues. Or, the pharma really knows exactly what target they want to go after. And they may even be working on it internally, but then there's something external.
There's multiple shots on goal. And what about the financial disparity? Because what I mean is, for example, a particular biotech's last valuation may have been $100 million, but the pharma will come in and buy it for $500 million to $1 billion. Whereas if on that exact same data, if that biotech were to raise again, there's no way in hell that would have been their valuation. So why do you think either it's like you could view it as the VCs are getting a great deal or the pharmas are overpaying? Like who knows what the true price should have been? But what do you think causes that disparity?
Yeah, that's a really interesting question, Armon. So I guess every asset's value depends on who wants it. And so from a biotech side, I guess if you can create the demand and the FOMO, the fear of missing out, then you can get a higher valuation. That's probably, I'm not saying it's all that, but that's significantly related to that. And then as you know, also in VCs and pharma, you look at comps. So usually then when you have a deal, the first thing people look at is, well, what were the prior deals in that space that would be analogous? And then you sort of start there. So that gives you the floor and ceiling almost from the get-go.
Well, I'd also maybe argue that there's an interesting dynamic because the VCs are kind of on both sides of the table usually because they do have their fingers already in that company. So they have like a mixed incentive of like, I don't want to pay too much. I want to leave because they're all aiming for an eventual pharma buyout or an IPO, but they kind of want that eventual pharma buyout. So they're like, careful about managing price to a zone where their next delta remains huge. Whereas often when you guys are coming in, you don't have your fingers. You as a pharma, you have no fingers in there. So it's like a true opposite sides of the table back and forth. So I don't know, I feel like some of that dynamic, I don't know what to do about it. I'm not saying I have a solution, but I feel like that can also lead to that. Cause I feel like I would always see these price disparities. I'm like, I know their valuation was that. Why did the pharma just say that?
I don't know if the pharma knows that, to be honest.
I don't think they'll realize it either.
Yeah.
Because I remember we were chatting about something and you were going to have a meeting. I'm like, do not tell them who you are.
Yeah.
You're going to walk in as William off the street who likes tennis.
That's true. That's true.
Sorry, and so now you've actually gone to our side of the house with the biotechs. What's it been like switching over? What surprised you from waltzing over to our side of things?
Yeah, I mean, I obviously in biotech, I'm the CEO of Revelio Therapeutics. You have to spend a lot more time fundraising. In pharma, I would get a budget and justify why I should get that budget. And I have to say I was able to get an increase in usually every year, unless there was like a big reorg or something like that. Whereas in biotech, I'm only as good as my next tranche. And then have to convince the funders that I should get the next tranche or, to the next raise and so on and so forth. So a lot of time is spent on that. What's interesting, I think, is that, the other thing is I roll up my sleeves all the time. And so I'm doing whatever it takes, right? I'm making slides, interpreting the data, having meetings with VCs, with strategics, and so on and so forth. So just whatever it takes, because I'm invested in the company.
Are there things you miss about the pharma side?
So let me come back. So I would say I used to get a lot of complaints, I would say, from teams about the amount of slides that they had to make in pharma.
Yeah.
But I would say I would have no mercy today, given the number of slides that I've had to make for the various meetings that I have. And it's been really interesting. I think the other thing that you see is like when you meet with a lot of these various investors, you get a lot of questions.
Yeah.
Whereas internal teams inside pharma, you don't really go outside of your group because you don't want to disclose anything.
Yeah.
So you're only meeting teams internally most of the time. And so, yeah, I'm just wondering, like, if I were ever to go back to pharma, like, how would you replicate that kind of real-time feedback? Because a lot of times the feedback is really good.
Yeah.
Like, and it keeps you on your toes and stuff like that. Now, inside pharma, you may have to go to the different functions meetings.
Yeah.
But it's not like the same, I think, kind of experience.
Well, for what it's worth, I don't know. I don't think it's that practical, but I do think the public side investors can be quite good. But I think it's just they don't normally want the R&D people to talk to them because they're worried about, are they going to be filtered enough to talk to a public markets analyst? Because I think that feedback mechanism does exist for like a public biotech still.
Oh, that's true. Yeah.
Which is like, almost all the pharmas are public companies. So theoretically, they can actually go do that too. I just feel like access is way more restricted by the pharma. The analysts would love to talk to the head of R&D.
Yeah. But I don't think my internal teams at Roche and Pfizer were going externally and meeting.
Right. They're normally
this
not
is
allowed
our program.
To.
What do you think about it?
Yeah. Normally they're not allowed to.
Yeah.
Because I think that feedback loop could exist. But that's an interesting point. There's a high risk of groupthink within a pharma.
Yeah, exactly.
Because there's so little outside feedback.
Stuff, right. So I think it's important for people to think about how to get that.
That's an interesting point.
Feedback. Let's see. What you asked me, is there anything I miss?
Yeah.
Well, I guess
aside from making less slides.
Okay, no. I mean, going back to what I said before, right? Inside pharma, there's an expert for everything.
Yeah.
So if you have a question, you don't have to go find, who am I going to talk to? Which consultant am I going to figure out? You just go to the person and be like, how do we deal with this issue, right?
Yeah.
Whereas now, I'm fortunate. I know a lot of people, but I still have to like get a consultant potentially.
Is there something that's much easier on the biotech side versus the pharma side?
Yeah, I think the speed of decision making. Yeah, for sure.
It's 20 less
say something this minute, and the next minute it gets done. Whereas in big pharma, it may take like another a month or something.
Yeah,
like
or if it ever happened.
Yeah, exactly.
And now that you've been experiencing it firsthand, do you see the VCs any differently on the biotech side? Because I'm sure you had some interactions while you were in pharma. Like, what seems different there?
Yeah, I mean, I think I had less direct interaction with the VCs, right? Because in pharma, we had BD teams. Yeah. And usually they would deal more with that. So I didn't have to consider as much, and I mean, we were more interested in interacting with the direct companies. Yeah. Of course, we would get updates from the various VCs on what's going on. But still meeting with the company. Yeah. Yeah. So.
Well, and partially I'm wondering it in the sense of, like, I feel like there's very often this pattern of certain pharma types will jump into biotech in C-level roles. Oftentimes they'd argue having no idea what they just got themselves into. So if you were having a talk with one of those folks, like, what is the part that you think is going to shock them the most?
Wait, say that again?
So there's often, like, I don't know, pharma BD or R&D people that get recruited into some C-level position in biotech. And I feel like many of them often have no idea what they just got themselves into. Like, I'm not saying they're not happy in the end or sad in the end. I'm just saying, like, it's often a shock. What are the things that you think tends to shock them most?
Well, I mean, I think if it's an early stage company where the science is so critical, I think the question is, would they be able to direct and manage the science in an efficient way?
Oh, so if they were like a, let's say, pharma BD person, and then now they're suddenly a CEO of a biotech, they're like, I need to, like, run science, and I'd never done that. Yeah. Or it's been years since I did that.
Exactly. Yeah. I mean, I guess, that's an interesting question. I have to think. Well, that would be the one way I'd answer that question, right? So if they were in BD and pharma, and then they were going to say like an early seed company, would they have the chops I'm not disrespecting. I'm
Yeah,
just saying I don't know what their background is, but if it's been more commercial or whatever,
Yeah.
or they've never run an R&D program themselves, then would they be able to do that? You obviously have to hire people that know what they're doing and stuff like that.
Right.
Yeah. I think the other thing is just the connections, right? You need to have, I guess, being in BD and pharma, they would probably already have the connections.
Yeah, because I think the assumption on, I don't know, both the hiring party and them making the jump is that they have connections, and when the end goal of biotech is very often a pharma buyout. Because even if they go public, they still want to get bought.
Yeah.
Very, very few of them end up actually selling their own drugs and everything and being successful. Very few. So they also want to be bought. So the assumption is, they know how pharma's buying machine works, so they can work towards that end goal. And they'll have some connections to assist the other things. But then I would guess operationally, they have no idea what life is like in this kind of raggedy, much less resource get shit done on your own.
I think one of the feedbacks I got also when I first started my company was, well, are you willing to roll up your sleeves?
Yeah.
Because you've been in big pharma, you have an army of people helping you.
Yeah.
Are you going to be able to schedule your own stuff? Stuff like that.
Move your own slide.
People forgot I had a lab, and labs, you have to be very scrappy as well.
Yeah.
In academia. So I guess that would be the same thing. If a BD person had a lot of people helping, would they be able to do stuff and roll up their sleeves? Yeah. The other one I think is, at least in my experience, some of the BD folks haven't had a lot of management experience. They're more individual folks. And so when you go to a company, could you manage?
That's true. Yeah, there'll be a lot of individual contributors. Having seen it from both sides, which not a lot of people get to see it from your vantage point, what do you think defines the biotechs that'll succeed versus fail? Or what do you think are the most common characteristics that will define they're more likely to pull through? Because most of them will die.
I still think that it's the science that should win. So do you have a really good scientific rationale and data supporting your hypothesis? Then should it be better than something that's out there?
My one challenge though would be
okay. Can you sell it?
Okay, that last part. I think there are cases that from a, let's say, financial success metric or acquisition success metric, you actually never knew if the thing worked by the time it got bought.
Yeah.
And people only found out after. And many of those cases, it actually didn't work.
Yeah.
So it ended up, I'd argue, having nothing to do with the science, but everything to do with the salesmanship. I know that's a little bit cynical, but do you think sometimes it'll happen that they just got to be really good at pitching the science, but not necessarily good at executing it? And that can be enough?
Look, I have to be staying in this industry.
That's fine.
Yeah. I think, ultimately what I would say is I would hope that people are making molecules that would succeed.
Yeah.
Mainly because, well, two things. One is, it's a lot of investment.
Yeah.
The second thing is you are putting patients on trials.
For sure.
And so I always have I call the grandma test.
Yeah.
Which is what you put your own grandmother on this trial.
Yeah.
And if you wouldn't, then at least for me, then you really shouldn't be developing that molecule. So I would just keep that standard for myself, at least.
100% agree.
Okay.
Cool. And then what do you think is going to be the next wave of innovation coming in for our sector?
Well, obviously AI is really hot right now.
Yeah.
And so...
which part of the hotness do you agree with, and which part of it do you that's overrated?
Well, I think we're in the hype phase for sure.
Yeah.
And I do think it will revolutionize the way we do a lot of things.
Mm-hmm.
Will it revolutionize everything? I'm not sure.
Yeah.
But certainly it will speed up some aspects. We see already evidence that, you could screen fewer molecules, for example, than you might have had to do in the past. Getting information, obviously. I think some of these new programs like AlphaFold, are really useful for novel ways of thinking. And then now there's many biotechs showing that they say they can design molecules from AI without having to do as much in the lab. But I would say that the serendipity part and the unpredictability of what happens actually when you get into humans, I'm not sure that we will address those with AI. And let me just give you some examples.
Yeah.
So like when the first EGFR tyrosine kinase inhibitors entered the clinic, though this is in the late 90s, early 2000s, we didn't even know about EGFR mutations. And they entered the clinic, and basically, even though there was a lot of data from the literature saying that EGFR was overexpressed in a lot of cancers, it should be applicable to a lot of different cancers. They only worked in 10% of lung cancer patients. And then after the fact, we found out they were EGFR mutations. Similarly, like alpelisib, which is a PI3K inhibitor, first of all, that was the fourth molecule from Novartis that made it. And the three other ones had died for various reasons, even though they had great preclinical data. One died for tox, one died for poor PK. And again, it's hard to predict when you get into humans. Now, I don't know, maybe AI can help improve that, but we'll have to see. And then also PI3K mutations were discovered after the fact. So there's lots of examples of you bring a molecule into a human and you might think it work one way, it will work one way, but like it surprises you, either from a tox perspective or from an efficacy perspective. And if AIs work based on prior knowledge, how is it going to address
well, that, and also, because I 100% agree, like the biggest cliff seems to be what happens when you put this in a person. And because all these models are as good as the data sets that train them, and I'd argue measure so few things in a phase 1 trial, because a lot of it is like, you don't want to know about bad shit that's happening. If you're not required to look at it.
Well, once you get into humans, you have to do that.
Well, I think it's more like there are certain things that they're like, I'm not going to specifically measure that. I'm just going to see if it pops up in my normal tox profile, and then I'll deal with it then. So the data is often not sufficiently mechanistic and or available. Because like Roche sits on its own pile, like Pfizer sits on its own pile, and so on. These aren't necessarily all connected to each other. So I feel like there's so little detail available when we are such insanely complicated systems. And therefore I'm like, how are you going to even train a model?
Well, that's what, for me, that's the biggest jump, right? Can AI address the going from preclinical to clinical? And right now we just don't have good enough faithful human model systems. Right. So there's 30 to 40 trillion cells in the human body. There's like, 20,000 genes. And I don't know what the latest in protein coding transcripts are. It used to be 90,000. I think it might be more. And then there's 8 billion people on the planet. So there's no model systems right now.
And all the chemistries that can happen because protein X and protein Y happen to be next to each other in this cell, but not in the other cell.
Or if you have a small molecule that has a metabolite that does so and so.
Exactly. I think it's so unpredictable, especially for small molecule. Biologics, it might be a little bit more predictable,
but other aspects of drug development, I think AI will be applicable to.
That'd be good to see. You definitely have your cool new book out, Breakthrough. What made you want to write that book, by the way?
Actually, I had a conversation with someone recently who said, oh, is this like the book you would have liked to have read when you took over the head of PMed? And actually, that was a really good way of summing it up. It's like, I wish I had a book like this before I actually did drug development. So I would know more exactly what was the process and what to look out for and stuff like that. So that is one reason. A couple other reasons. I wanted to highlight the unsung heroes of drug development. So we talked about academia and industry. In drug development, a lot of the academics, and again, they deserve it, but they get a lot of the credit for the initial discovery. And of course, we wouldn't have a molecule without the initial discovery. But getting from an idea to an actual therapeutic is a whole nother level of work that is required, that requires a whole nother level of work. And there's a lot of scientists that work on those, and there's a lot of failure that goes into that. So I do want to highlight like the chemists, the toxicologists, the clinicians, all the people that sort of contribute.
Well, that totally resonates, because my exposure, I guess, to that part was when I was a grad student, then I went to be a postdoc, and I was starting to, I don't know, when you're a naive student there, you're like, oh, we do the hard part, the hard but glorious part of coming up with these targets or molecules or whatever. And then some lower life form can do the translation piece because we did the intellectually difficult part. But then the part that was making me really question that / want to jump to the other side, I was like, oh, there was this like Nature paper that looked awesome. And then I'll go ask my PI, because I worked for some cool people. Like, I don't know, I remember asking Uli von Andrian, who was my immunology postdoc supervisor, like, oh, what happened to that paper? He's like, I don't know, we published it, postdoc left, that's that. I'm like, okay, but Yeah, like no translation. And I think he had a very good point. He's essentially like, look, our current, I'm paraphrasing, but our currency is papers. If I do the next improvement or make it more drug-like, that's not a Nature paper.
Yeah.
Like no one cares.
Yeah.
So my next big paper is an NEJM paper, which is at least $10 million in a few years away. And we as academics are just not set up for that.
Yeah.
So no, those projects are just going to sit there unless pharma or someone else comes and gets it.
Yeah.
And we just move on to the next thing and hope that something happens with it. I'm like, yeah, okay, I can't do that to my project.
Yeah.
I got to jump.
Well, in the book, we interviewed David Altshuler.
Yeah.
A great quote about, in academia, you're building a bridge with Lego. Again, no disrespect.
Yeah.
But then getting a launch medicine is like building a bridge across the Hudson, right? It's like a whole nother scale.
Yeah.
And also, I think appreciating that even manufacturing has its own scientific expertise. We tend to maybe sometimes think it's less relevant, but actually being able to synthesize and manufacture a molecule at scale that can be given around the globe safely, and so on and so forth, takes a lot of effort.
Oh yeah,
which I think is underappreciated in medicine.
Yeah, well, because at the lab scale, you never deal with that. You know,
Exactly.
make your tiny batch for your mice or for like 10 patients. It's very different from what you said.
Yeah,
to make it consistent. And like one of the other things you were mentioning, you mentioned in the book, is around this whole institutional memory where like they learn lessons along the way until years later they kind of figure it out. Is that part of what you were referring to as well, with like it turns out it was a genetic mutation we didn't know about?
Yeah, I mean, well, in sort of institutional memory, so the book is about 8 different molecules from 8 different companies. And I think what's unique about it is it not only starts with sort of the initial clinical description or the academic discovery, but then it actually shows what happened inside pharma or biotech to actually make that medicine. Right. And that's rare because I got to interview many of the scientists inside these different companies. And the best example of institutional memory actually is the chapter of the Paxlovid, where basically the Pfizer team had been working on the original SARS virus in the early 2000s and had already thought the 3CL protease, the target in the virus, was relevant. And so they had a whole team working on it. And then when the virus disappeared, the molecule got shelved. But then say, what was it, 10 years later? 10, 15 years later 15, 20 years later, when SARS-CoV-2 came around, there were like a few people left, even though the infectious disease group was disbanded. There were a few people left that were like, oh, we worked on that thing many years ago. And actually they revived the program and then had a jump start sort of on getting there. As
that was super cool.
So I think the lesson there is, the other thing, many people in pharma and biotech can work on molecules and drug development and never have a molecule succeed. But I think the lesson is if you're working hard every day, you understand the principles of drug development, and you remember your lessons learned as you go along, then someday serendipity may fall into your lap and you may be successful. But if you're not learning along the way, you won't be successful.
And in the meantime, they're going to shake their fists at the head of R&D that killed their program.
Exactly.
So if you could wave a magic wand and change like one thing about how pharma R&D works, what would you change?
Oh, that's a tough question.
Maybe you're allowed to change 2 things. There's just limited magic in this wand.
Well, yeah, I mean, okay, unlimited resources. How about that one? That's an easy one.
Well, maybe joking aside.
Although I would say having limited resources also is very good.
What do you think would drive more resource into this? Because I think an obvious comparison right now is like the insane amount of money going into AI stuff is just an absurd scale compared to what pharma is used to. On the other hand, I'd argue on average, people would rather, not die or live a healthier life than, I don't know, scroll their apps or have better purchasing suggestions for their next widget. So why do you think there is that delta in how much money will go into, we all use it, but relatively dumb things? Well, no, but it's more like hundreds of billions going into, let's say, AI type R&D is just a thing, but it's definitely not going to happen in pharma, at least not for now. But what would it take for that to happen? Because people clearly care about health problems.
Yeah, I mean, right now, since 1945 in the US, the way the whole system has been working is that government has invested in basic science and curiosity-driven research.
Yeah.
And so I think the NIH budget's like 50 billion.
Mhm.
And, I think pharma is not necessarily going to do that. I don't think the VCs will do that.
Yeah.
Because the payout is too far away.
Yeah.
You could be working on a problem that has no therapeutic relevance,
Mhm.
or you could be working on a problem that like all of a sudden you're like, oh my God, this has amazing therapeutic relevance, right? And there's so many examples of that. Like even CRISPR was like being worked on in bacteria.
Yeah.
Right. And then once Jennifer Doudna and Emmanuel Charpentier figured out the mechanism of CRISPR and how to make it applicable to mammalian cells, like, then like you're off to the races in terms of the therapy. So,
hence the no research, no cure.
Exactly. Yeah. So I think, I guess, well, I'm not answering your question, but I think I am concerned about,
not getting more research
are we going to continue to have the investment in basic science research in the US at least?
Okay. And because you've been guilty of being on both sides now, what would you change about the biotech ecosystem if you could change one thing? You don't get to say resources again.
Yeah, I guess it would hopefully be easier to raise money. No, you just said
what would improve the productivity side? Because obviously like throwing more money with productivity constant, it'll obviously produce more. Well, like what are some of the inefficiencies
what
you've
I've
noticed?
seen is that the VCs, they get to see everything very early on. It's quite amazing.
Yeah.
From the seeding the company to the the preclinical to the phase 1 data, they see earlier than anyone else. I think, and so maybe figuring out how to make that system more efficient, and opening it up more.
So that more like pharmas and others can see it too.
Yeah.
That's a good one. Okay. Cool. Any other final thoughts before we wrap?
No, I want to thank you for the opportunity. It's been a lot of fun.
Hey, thank you for coming. Appreciate it.
All right.
Cheers.